Welcome my dear students. Today we will discuss about the binary fission of bacteria. A mature bacterium divides into two daughter cells. These two daughter cells are identical to each other. Again, after maturity, these two daughter cells, they also divide. Thus, when the conditions are favorable, the number of bacterial cells as well as the mass number doubled. This is termed as binary fission process used for bacterial reproduction. The time gap between the two successive generations of binary fissions is termed as doubling time or generation time, and in bacteria it is generally 20 minutes if all the conditions are favorable. Now we will calculate the number of offsprings in a specific time, and for this process we have to use a formula. That one is 2 to the power n. Here n is the number, it may be 1 2 3 4 5 6 like that, and generally n is calculated by total time divided by 20 minutes. If the specific time is 4 hours, then total minutes is 240 minutes, and n will be 240 divided by 20 is equals to 12. Okay, so the total number of offsprings will be 2 to the power 12, that is 4096 in 4 hours. That is, we may say that if all the conditions are favorable, then a bacteria will be converted into 4096 bacteria cells by means of successive binary fissions. So this is how the total number of progeny or offsprings may be calculated. Now the process. The first step is DNA duplication. At first, two strands of DNA replicate, then divides into two equal halves. So the second step is DNA partitioning. After DNA duplication, each one of the double strands swings towards one of the cell halves. Thus, each cell hub receives one double strand of DNA each. Then, cross-wall formation. DNA doubling is then followed by inward growth of the cell wall at midway between the daughter genomes. For this, cell wall materials deposited between the membranes and the cell is divided into two daughters. The process has advantages. The first one is only one parent is needed in binary fission. And the second one is it is a rapid process. For example, one E. coli divides in each 20 minutes. The third advantage is daughter cells are identical to the parents. And the fourth one is many daughter cells are produced in a short time. For example, one E. coli produces 4096 daughter cells in 4 hours only. And regarding the disadvantage, the cells produced by binary fission cannot survive in changed environment due to the lack of genetic recombination.